The second part of blood buffers, the chief buffer systems. There are bicarbonate buffer system, hemoglobin buffer system, protein buffer system, phosphate buffer system. Let us see what are the contribution made by these buffer systems to the entire buffering capacity of the body. Now, bicarbonate is the leading one with more than 50% of the buffering capacity of the body is taken care of by bicarbonate buffer system. Then hemoglobin is there, 35%, and small contributions from protein buffer system and phosphate buffer system. So let us go to the prime one, bicarbonate buffer system. The most important buffer system on the body. This is often H2CO3, HCO3 minus. That is carbonic acid bicarbonate buffer system. Now, this is often called an open buffer system because it exists in two separate compartments, which we will see in a moment. And carbonic acid here is the acid component. It's a weak acid. It's an acid because it can release H plus or donate protons. And HCO3 minus bicarbonate ion is the base. It's a conjugate base of carbonic acid because it can accept H plus and become H2CO3 again. Here, it is seen that the ratio of H2CO3 to HCO3 minus at normal pH of 7.4 is 1:20. As you can see here, it is 1:20. Means that for every single part of H2CO3 carbonic acid, there will be 20 parts of bicarbonate ion. Here, we may expect a ratio of 1:1, but it's not so. It is 1:20. Instead of 1:1, and the HCO3 here represents the alkali reserve. Obviously, it has to be sufficiently high to meet the expected acid challenge. And as we had discussed earlier, body, our cells are chiefly challenged by acids and acid load. Now, if it was 1:1, all the bicarbonate ions would have been exhausted within a very short time, and the buffering capacity will not be very effective or very proper. Therefore, under physiological circumstances, the ratio of 1:20 is very crucial, and it ensures high buffering capacity and high buffering efficiency of this particular buffer system, namely bicarbonate buffer system. Now, let us see another equation like here. This equation shows how carbonic acid is formed primarily. It is actually formed from carbon dioxide and water, and you can see that both these reactions on either side of carbonic acid, both of them are reversible here. And the issues on this part, on the left part, will have respiratory cause because carbon dioxide is dealt primarily by lungs, and it is under the control of lungs. The respiration events, that is the expulsion of carbon dioxide, is under the control of lungs. On the other hand, the issues on the right side, this part, will have something to do with the kidney and have some metabolic causes because bicarbonate ion is primarily under renal control. Let's examine a few conditions on the left side and the right side. Now, here, if H plus level in blood is high, so high acidity, a condition called acidosis. So during acidosis, the excess H plus ions combine with the bicarbonate ion to give H2CO3, the acid component. So this is acid component, and this acid component is under respiratory regulation. This can split to give carbon dioxide, and carbon dioxide is expelled by enhancing respiratory rate, or hyperventilation occurs. The chemoreceptors in brainstem, medulla oblongata, it has got chemoreceptors which can detect changes in pH in blood and regulate the respiratory rate accordingly. So during acidosis, respiratory rate is enhanced so as to expel more carbon dioxide. See, on the other hand, during alkalosis, the respiratory rate is reduced. Hypoventilation occurs. This occurs. And carbon dioxide is withheld, so it can get converted to carbonic acid, and H plus is contributed. so that during alkalosis, respiratory rate is reduced. So that ultimately it will give rise to production of more H plus ions. Now, on the other hand, let us consider the right side, this part. If the basic component HCO3 minus bicarbonate ion is high, now which is called alkalosis. So during alkalosis, kidney remove HCO3 minus through urine. It removes through urine, which means kidney regulates pH by excreting alkaline urine. And at the same time, kidney will simultaneously retain H plus also. It can retain H plus also. And it holds H plus and do not allow H plus to be excreted. But during acidosis, what happens during acidosis? Kidney reacts differently. When blood acidity is high, kidney efficiently reabsorb almost 90% of the HCO3 minus, which is passed through primary urine. And together with it, it can excrete excess H plus also. Which means excrete H plus ions, and H plus ions are excreted actually as ammonium ions, NH4 plus ions, and during this period kidney excretes acidic urine. So what we have discussed so far has been written here, described here. Can go through the lung mechanism, the kidney mechanism, then the respiratory mechanism. What happens during acidosis, alkalosis, the hyperventilation and hypoventilation occur, as well as renal mechanism is also written here. Now we saw that bicarbonate ion can be released or reabsorbed. H plus ions can be retained or excreted. Now as well, this third condition, bicarbonate ions regeneration. So that is in the DCT of kidney. It is a mechanism called renal glutamine mechanism, which we can see very briefly. The new bicarbonate is generated, how it is done. You can see here glutamine, it's an amino acid. It gives rise to glutamate, another amino acid, and during that time ammonia is released. And ammonia can take up H plus ion, and become ammonium ion, which is excreted. So acidic urine is excreted. Now at the same time, what here formed as alpha ketoglutarate. Alpha ketoglutarate is a member of TCA cycle. It can give rise to carbon dioxide through TCA cycle, and carbon dioxide can be converted to H2CO3 and then HCO3 minus. So bicarbonate ions are regenerated, or new bicarbonate is generated here. So this is called the renal glutamine metabolism. Now let us examine some of these conditions. This is acidosis and alkalosis, some disturbance in pH level. The normal H2CO3 HCO3 ratio will be sometimes upset, giving rise to acidosis and alkalosis. Now the first two, metabolic acidosis and metabolic alkalosis, are concerned with the level of bicarbonate ion, and next to respiratory acidosis and respiratory alkalosis are concerned with the level of carbonic acid and carbon dioxide. The first one, the metabolic acidosis. Metabolic acidosis is caused by deficit of bicarbonate ion. It is during that time pH level is decreased, which means it is less than 7.4. And this is usually most common cause of metabolic acidosis is diabetic ketoacidosis or diabetic ketosis, when ketones and lactates are enhanced in blood. Now, metabolic alkalosis, when pH level is increased, alkalosis 7.4, more than 7.4, that is pH level. And this happens when excess bicarbonates are there in blood. And this may be a result of excessive vomiting. Excessive vomiting when H plus ions are lost excessively and HCO3 minus ions are increased in blood. And also during administration or over ingestion of bicarbonates ion. As a part of therapy, this bicarbonates may be administered. So during that time there is chance for excess administration, and which result in metabolic alkalosis. And here respiratory acidosis, when the pH level is decreased, that is less than 7.4, it is decreased. This is due to excess carbonic acid. Excess carbonic acid means carbon dioxide retained And this may be a result of, mostly a result of obstruction of respiration, as happens during some conditions like emphysema or asthma. And finally, respiratory alkalosis, when pH level is increased, that is more than 7.4. There is a deficit of carbonic acid, deficit of carbon dioxide in blood, and this is usually by hyperventilation. Now, when this occurs during when some people reaches high altitudes, all of a sudden they can have this what is called hyperventilation, so as to adjust the pressure, high pressure, and the high altitudes. So these are some of the conditions that when H2CO3, HCO3 minus level is upset. Now let us go to the other kind of buffer systems now. So hemoglobin buffer system, this accounts for more than 35% of the entire buffer capacity of blood. Now, the buffering capacity of hemoglobin depends actually on the presence of COOH and NH2, obviously the amino acids present on it. Here especially important is arginine and histidine. Both of them are basic amino acids. Here this nitrogen, the imidazole nitrogen of histidine, is very important in that it can bind oxygen through iron. So ferrous ion, Fe2+, ferrous ion is attached to this nitrogen, which in turn accepts oxygen. So this histidine is very, very important in buffering capacity as well as functioning of hemoglobin as a carrier of oxygen. Now, let us see how oxygenation of hemoglobin affects the buffering capacity. The most important thing here with the hemoglobin is that when it gets oxygenated, it becomes more acidic, and when it deoxygenated, it becomes less acidic. Oxygenated, more acidic; deoxygenated, less acidic. In lungs, what happens is that when it accepts oxygen, the HHB releases H+, and this H+ is actually accepted by bicarbonate ion that we will see in a moment. But in tissues, it gets deoxygenated. So because oxygen is unloaded from hemoglobin, and when it gets deoxygenated, it accepts H+ and becomes HHB. Now, what is the role of bicarbonate? The bicarbonate, it accepts the H+ release from HHB, and it becomes H2CO3, which can break to release carbon dioxide, and this carbon dioxide is released to plasma and expired through lungs. So these all things happen in lungs. The HB becomes oxygenated, it becomes acidic, it releases H+ from HHB. This H+ is accepted by HCO3 minus, it becomes H2CO3, it releases carbon dioxide, and carbon dioxide is expelled by expiration through lungs. Now, the entire thing is reversed in tissues. In tissues, the HB gets deoxygenated. It releases oxygen, it unloads oxygen to tissues, and at that time it becomes HHB. So whatever happened here in lungs, the H+ released becomes HCO3, carbon dioxide is released through lungs, and in tissues the H+ is actually the carbon dioxide is accepted from tissue. The entire process reverses, and H+ is accepted by hemoglobin to become HHB. So this is how hemoglobin it functions as a buffering system in blood. You can go through this the entire process and study it yourselves. Now we move over to protein buffer system. Now proteins have obviously they have so many amino acids in them, and the buffering capacity of protein actually depends on the ionization of amino acids in it. Like you can see here, the citrion form of an amino acid. In acidic medium, it can accept, the citrion can accept here, the COO minus can accept an H+ and becomes COOH here, like here, and it becomes a cation at low pH, when the medium is at low pH, or H+ ions are available. On the other hand, if H+ ions are not available, or the medium is in high pH, the COO minus remains the same, but the NH3 part, the NH3 plus part, this part, it releases H plus to the medium and it becomes an anion like this. So COO minus, this anion, this imparts anionic properties to this part. So this is how a protein basically act as a buffer system. Now finally, phosphate buffer system. In human body, the phosphate buffer system is used as an intracellular buffer system. Now phosphates are found in two forms in blood, namely sodium dihydrogen phosphate, NaH2PO4, which is the weak acid component, and disodium monohydrogen phosphate, Na2HPO4 minus, which is the conjugate weak base of this buffer system. Now you can see how this is formed and broken down here, and the ratio of NaH2PO4, the acid component, and Na2HPO4, the basic component, is 1:4 with the basic component dominating. Now, you can see when blood is acidic, this part, Na2HPO4, it accepts H plus ions and becomes NaH2PO4, and this acid phosphate is excreted through urine, making urine more acidic. On the other hand, when blood is alkaline, H plus is released in plasma, and this part, the basic phosphate is excreted through urine, making urine more basic or alkaline. So that is how phosphate buffer system works. Thank you.